Yeah, so I am currently a lecturer at Sturt University with Wagga Wagga. And so I've recently taken up that position, and I usually leave out the Canberra bit because that's too confusing. So before I joined Sturt, I was a postdoctoral researcher scientist at WEHI for the past 5 years. And so I'm still honorary research associate with them. And Ian, I'm not sure how much mammoth there will be in my talk today, but it's quite fascinating to think about that. And there's a new Jurassic Park movie coming out as well, I think. And yeah, it would be great to have a conversation maybe after or at some point. Yeah, so I've put my email address there in case anyone has any questions. They can always send me an email. And I thought sort of already have immediately like some picture to sort of set the scene on my slide, where so obviously CRISPR is this like is an abbreviation for a very complicated term that I won't use because yeah, mostly it's like CRISPR sort of known as the new gene editing technology on the block. And so when we talk gene editing, genome editing, then it's all about DNA. And so we all have letters of DNA in our cells. So these are our genetic code. So they start with these, we have these 4 letters. So A, G, T, C. And so that's basically like a book, and we have sort of different letters making up genes. And if we would write all our billions and billions of letters, then they become like too big really to print it. And so what cells do is they have it all packaged up in what we call chromosomes. So you can see on that picture already how it's sort of packaged, how the DNA is going to be packaged in sort of really compact chromosomes, and we have 46 of those chromosomes in every of our cells. So before I keep going, I just wanted to also take the opportunity to really highlight the country and the lands that we're all sitting on at the moment. So we all, if you're in Australia, that is, we all sit on traditionally owned land. So before English people came to Australia, someone was living here already. And so I think it's very important to just reflect on that fact. And so I'm currently in Gadigal country, which is Sydney. When I'm in Wagga, where I live, that's Wiradjuri country, and Melbourne is Wurundjeri country, Canberra and Ngunnawal country. And so Wagga Wagga, where I currently live, is also like, if you engage with the language as well, Wagga Wagga means the place of many crows. And I think it's just really important to reflect on that and to really appreciate the rich culture that First Nations people bring and how much we can learn from that. And so I have this slide here from the National Reconciliation Week, which just started yesterday, I think. So yeah, it's really time that we all help to bring about some change. Right, so then I thought I'd just have a quick overview of my talk. So what I thought I do give an introduction into gene editing and also CRISPR technology. And then I sort of give a few examples of how I use it in my research. So I use it for disease modeling or identify novel disease targets. And then also at the end, I just sort of show how we're going to implement it for a new sort of rapid diagnostic tool. So I can't promise that my language might not be too much jargon or too advanced for one or the other. So I just wanted to highlight as well. So I also so a lot of the pictures that I have in my course, in my slides here this afternoon, is from a online learning course that I've designed while I was working in the education office at WEHI. So I had a bit of a secondment during COVID while lab work was sort of shut down for a while. So we came up with this online learning course that's freely available for anyone. If you go to that website at the bottom, we called it CRISPR 101, which really gives you all the basics, everything you always wanted to know about CRISPR, starting with sort of what is DNA and then leading into where CRISPR comes from. And we have a little bit of that in the talk as well. But if you, yeah, really encourage you to check out that course. And so, So if we go through the first part, which is sort of introduction, so then I like to start with the sort of the DNA, code of life, where we have our 4 letters, and this is our genetic makeup. And so in the end of the day, what DNA codes for these are sort of proteins that function or then have a function in our cells. So in our mouth, so I'm speaking now, so we have saliva, and in that saliva there's an enzyme called amylase, which sort of, as soon as we eat, that starts digesting food. And so every enzyme, every protein is encoded by these 4 letters in our DNA. And so that's sort of the background. And you can imagine that if you would change one of those letters, then that might have an impact on the protein that it's coding for as well. And so this is where then genome editing comes into play, where we can try and basically modify that code. And so we can cut things out, or we can add things, we can repair things. And so this is usually what you can use it for, is like for repair or to study. So for the repair aspect of genome editing, that would be something like gene therapy. You would find it in a clinical setting, where if you have a genetic disease or a disease caused by defects in genes, you can use genome editing technology such as CRISPR to try and repair that faulty letter and sort of put the correct one in. And what I'm mostly doing is preclinical research. So we're doing basically study the genes, study the DNA using CRISPR. And just have so CRISPR is relatively new. It's only like a decade or so now that scientists are using CRISPR. Before that, people were using something called homologous recombination. And I have this loaf of bread here just to show that it's from a lot of these molecular biology tools. They actually have been studied in yeast. And so yeast is a very important organism for us, because we can study a lot of things that we then can apply for the human situation. And before CRISPR, what we did with homologous recombination is we have sort of similar DNA molecules that recombine to create a molecule that we wanted to sort of generate, in case if it's like a new, if we wanted to repair something, then we can sort of have similar sequences left and right of that faulty defect, and then hoping that the machinery will recognize the similar sequences and then integrate it. And so just for very advanced people, just having some strategy, which we call like gene targeting strategies, which we published in these papers, where we use that concept of homologous recombination. And then also still before CRISPR, but also during CRISPR. So when we do genome editing these days, we use something called nucleases, which are genetic scissors, and what that means is they cut DNA. And DNA has 2 strands. And so this abbreviation there, DSB, means double strand break. So we use nucleases to cut the DNA at a precise location. And that is our start. That's always the start for our genome editing. And so before CRISPR, we had something called zinc finger nucleases and talons. And so they were working quite nicely. They were very laborious to generate, but they did their job. And talons, actually, they were just in 2011, they were just about to be used quite widely. But this is then when CRISPR came around, and I started saying only CRISPR, but the actual genetic scissor that we use is called Cas9. So it's like Cas means CRISPR associated protein. So, and Cas9 is the one that we use for gene editing. And by labeling it 9 sort of gives already away that there might be different Cas scissors if you want. So Cas9 is the one that we use for genome editing, but then just give at the end for diagnostics, just an example of how there are others with other numbers, Cas genetic scissors. So basically, if you only make it 2 messages that you take from that talk, so then make this one of them. So when we do genome editing, we rely on that cell that houses the DNA that we cut to help us with the editing. So our editing creates a double-strand break. So we cut the DNA at a precise location, but the cell doesn't like that. Like, it's very toxic for a cell to have the DNA broken. So if it can't repair the damage, then it will say, oh, no, that's too much. We have to abort. We can't live with DNA damage, so we have to abort. And so therefore, the cell immediately wants to repair it. So as soon as we cut it, the DNA wants to repair it. And so we use that to generate models of disease if we want, or models of whatever gene we want to study. So often when we want to study a gene, for example, what we do, we remove that gene altogether. So then if you don't have the gene for amylase, for example, then you might not digest the food in your mouth as much as you would if it's there. And so you can do that by just cutting the DNA, and then the cell wants to repair it. But the problem is that the cell does a very sort of quick job, like it has to repair it really quick. And so that is really error prone. And so you might gain additional letters in that position, or you might actually lose some letters. And either way, that process usually creates sequences of DNA that weren't there before. So even though it's repaired now, it's not the correct sequence anymore. So usually that means your reading frame for that gene is disrupted, and you basically get the net result is a knockout of that gene. And then for our other more precise editing, where we can edit a particular sequence to our choice. So let's say there is actually like a faulty letter at that particular position where we cut it, we still have to cut it. And then what we then do, we try to hijack that repair machinery that the cell automatically puts in place by providing a repair template. So we use something like something that the cell then can use to integrate at that position where we cut it. And so if we're lucky, then we provide that sequence that we want to integrate at that particular position with the correct letter, DNA letter. And so then we cut, and at the same time, the cell has that correct sequence that we want the cell to integrate ready at hand, and hopefully then integrates that. So that there are these two approaches, which all start with cutting the DNA, and then we rely on the cell to repair. So either on the left-hand side, we have this error-prone way, and in the right-hand side, we have the precise way if we help the cell by providing the correct sequence. And so this is usually how we do genome editing with CRISPR-Cas9 technology. And so this is the second take-home message, which is sort of the really nice feature, the nature of Cas9, where Cas9, in contrast to the previous genetic scissors that have been around, Cas9 is guided by a GPS system, if you want, that is made of RNA. And so I've put this table together just to compare the two mostly used tools, genome editing tools before CRISPR. And so I have the genetic scissor, or called nuclease, is Cas9. And the way Cas9 is finding the correct sequence to cut is by a RNA molecule that we can program. So we can design this RNA molecule and thereby then direct Cas9 to wherever we need to make that cut to edit. And the RNA is very easy to generate. It's very easy to reprogram. So there's a lot of, this is where the biggest advantage comes from using CRISPR-Cas9. Because the other two previous genetic scissors all rely on a sort of fusion protein where other proteins recognize certain sequences in the DNA, which is sort of very laborious and also not really that specific. So with RNA, is really like the RNA guided Cas9 system really is like revolutionary. So that's why CRISPR sort of skyrocketed in recent years and became sort of headline news at times as well. And so just highlighting in sort of more detail the components now, where you can see in this gray sort of shape is the Cas9, and then you have on the left and right your DNA, and we have this RNA molecule, which we call guide RNA. So GPS guiding the Cas9 to that particular position where it then cuts the DNA. So so we have these 3 components basically where Cas9 can bind to the guide RNA, and the guide RNA then leads Cas9 to the correct position in the genome to cut. And you see 3 letters PAM there, which is a requirement which we might see later as well. So this is just indicating 3 specific DNA letters. But I might leave this for later if someone wants to ask a question about that. But basically you need Cas9 and the RNA to guide it. And so just for scientists, this is our, you can imagine we have billions and billions of letters. And how do you then design a guide or guide RNA for then Cas9 to go to a particular location? And so there's software, there's algorithm where you can sort of have you tell basically a gene that you want to target, and then the software will find this 20 base pair guide RNA sequence. So our guide RNA for CRISPR, for Cas9, is usually 20 base pairs long. And so you would have to design it in a way that these 20 base pairs, which are made of A, T, G, C, that they are so unique that they're only occurring once in our billions of bases that we have. So it's quite mind-blowing if you think about it, how specific it would be. And then just sort of highlighting that there's a lot of technology development. So what I described is this traditional sort of using Cas9 to create a knockout, remove like a gene to remove its expression. You can do the opposite where, and I'm going to show this later as well a bit, where you can activate gene expression by having Cas9 not cut anymore. So you can sort of mutate Cas9 so that it doesn't cut, but still binds to a particular location. And you can also do it in the same way to inhibit gene expression. So you can use it to activate gene expression or to inhibit gene expression. Then we have a thing called base editors. They're quite hot topic at the moment, and prime editing as well. And so we look at base editing and CRISPR activation in a bit. So then applications are huge. You can use it for a lot of things, maybe like conversation of memos as well. So I usually, so you can, lots of clinical applications, especially if you have a disease that's caused by a faulty gene. So you can sort of treat the genetic condition with CRISPR, and you can also use it for, or that's about sort of being developed, that you can use it for diagnostics. And I usually use it for sort of traditional preclinical research where we do disease modeling and try to find novel disease targets as well. And just a bit more background is sort of included that here, where CRISPR is actually coming from bacteria. So most of the things, I showed yeast, so most of the things come from yeast or bacteria, and then they are very smart scientists that then repurpose them for sort of our usage, if you want. And so quite often they then are being awarded a Nobel Prize. And so one of these inventors is Jennifer Doudna, and she received the Nobel Prize in, I think, like 2020, not too sure, 20, yeah, it must have been 2020, I think, for discovery of CRISPR for genome editing. And she had this quote that science is a serendipitous endeavor. And that what she means is that she wasn't actually trying to invent the next genome editing tool. She was just like sort of researching bacteria and looking at their immune system. And so we have immune system which is like very complex. We're like complex human beings. We have like lots of T cells, B cells, a lot of immune cells floating around, but bacteria, and so on that picture above, there's a bacteria cell which is sort of in this brown color, and bacteria are also attacked by viruses. They're called phages, but what the phage is doing, it's inserting its DNA into the bacteria. And that's basically not good for the bacteria, because then the phages will reproduce inside the bacteria and sort of destroys the bacteria. And so this is what people were studying over time, and they termed this coined the term CRISPR in 2002 already, and then in 2005, Cas9 was identified as genetic scissor, and then 2011, sort of more sort of the individual components were discovered, and then really from 2012, 2013, is when people realized, hold on, we can use that CRISPR-Cas9 system to actually have a very specific gene editing tool in human cells, if you want. And so now, fast forward 2022, CRISPR's technology is used in more than 50 clinical trials, so mostly associated with single gene diseases such as sickle cell disease, some cancers and blindness, and also CRISPR Diagnostics. And so it's so famous, if you want, that my mom in Germany sent me that article from a German local newspaper about CRISPR and the implications of CRISPR and how quickly everything developed, where scientists went from how is it like the bacteria use it to defend against invaders to a really specific gene editing tool. And so just highlighting here bit of an excursion where, again, we have it from bacteria. Bacteria have a circular genome, and in that genome, if you sort of zoom in, you see that there's this Cas9 gene, which is the genetic scissor, and then a bit further downstream, we have something called the CRISPR locus, where we have all these sequences from that are originally from the phage. So that bacteria are very tricky, they're very sort of very intricate, where as soon as what we saw, the phage is putting the DNA in, but then the bacteria, as their defense mechanisms do, is they sort of chop this up and integrate it into their own genome, so that then they know what the phage looks like when it comes again. In a way, it's sort of what we do when we get vaccinated against a certain disease. So then we sort of know what it's looking like, and so next time it comes, we know what to look out for and can immediately sort of attack it. And then scientists basically took these individual components, put it in a test tube, see if it worked, and yes, it worked. And so I'm sort of fast tracking a little bit, where just in the interest of time, where we have the original CRISPR GPS system, the RNA, the guide RNA, had two components, so CRISPR RNA, trace RNA, but it's now together fused as one single guide RNA. And so yep, scientists, as we saw on their timeline, were able to show that it's all working together. And so we have then basically, if you add the genetic scissor together with the GPS RNA molecule into cells, we can get precise editing. And then, so I just put the slide with the CRISPR babies. So ethical considerations. So few of it in 2018, CRISPR babies were sort of reported, which is not good, obviously. And so we still have a lot of technology development to do with CRISPR. So, and also really, this is a good example, similar, some actually made the example with like a nuclear bomb scientist and society need to work together to address challenges. So you create a new technology, then it's there now. So what do you do with it? So it's not up to the scientists alone. So it's like a whole society has to sort of understand what we have here and sort of come up with a plan, what we do with it. And so I know I'm like a bit running short on time, so I'm just trying to see. So my research, or at WeHi, what we were doing is really looking into cancer, mostly blood cancer. And there's all these different hallmarks of cancers. And one of them is how cancer cells are not dying. So that's one of the hallmarks. So they keep growing. And so they keep growing at positions where they shouldn't grow. And so we were looking at why are they not dying? And using CRISPR is just mesmerizing how you have this pool of DNA and how it can be so specific to have a precise cut, double strand break at a precise position. And so having our sort of blood cancer background, this is sort of what we do this double strand break, which we then rely on the take-home message one. We realize that we really need the DNA repair from the cell to then either have this error prone, so NHEJ, or like a homology directed repair with a precise editing if we provide a template. And so with that, we at Melbourne, at WeHi, I was part of the Melbourne Advanced Genome Editing Center for MAGIC. And so we generated all kinds of disease models depending on particular genes for WeHi or for the wider community as well. And I just skip over these examples, just ignore all the clicking, but that's just an example where we use CRISPR to remove a whole gene, so all the different exons, by using two guides if you want, and everything in between is cut out. And Another example is where we were able to introduce a specific mutation, the C14R, so changing an amino acid by changing one letter in the DNA. And for that, we cannot just cut it. We also have to present that template with that correct mutation or sequence that we want the cell to integrate. And so we sort of did the model, and and it's just some validation of that model. And then I just want to highlight a lot of the focus for me is about finding novel disease targets. And we have billions of letters of DNA. We have 20,000 genes. And a lot of, if we have particular disease such as cancer, then there's a lot of genes that are potentially responsible for driving that particular disease. And often it's a little bit like finding the needle in a big haystack. So, and CRISPR is helping us with that. And just going back to the background where we do this sort of new target gene discoveries, like modeling human disease. And there's a good model that we use for Burkitt lymphoma, where we have translocation from the CMYC gene from one chromosome to the other, and that is driving the lymphoma development. And so we can use those lymphoma cells and actually do a big screen where we screen the whole haystack, and we call that like whole genome screening. So we first make sure that we have the genetic scissor in the cell, and then we put all guide RNAs corresponding to all genes. So we have 20,000 genes. Then we put often 5 guides per gene. So we have like 100,000 of those guides that we add to the pool of cells, with the hope that each cell has one particular gene removed. And we then add a drug which kills the cells. And if a particular removal of a gene helped the cell to overcome that drug treatment, so if it's like a chemotherapy drug, usually all cells should die off. If a cell has acquired this survival mechanism by not having that gene anymore, then that cell would be surviving. And this is then where we then go back to the DNA and check what is actually going on there. So we use sequencing and find out what gene there was responsible for that cell to survive. And so that is published in bioRxiv at the moment. So we have this whole genome screen, and just skipping through that a little bit as well, but we did a similar thing where instead of removing a gene, but cutting it out, we can actually make more of that gene. So we do an overexpression, and we use that BCL2 gene, which is important in blood cancer. And so instead of cutting DNA, we have this version of Cas9 which can't cut anymore, and we fuse it with like activators of gene expression. So there's transcription factors that usually bind to promoter regions of genes. And if we direct Cas9 to that using the RNA as a GPS, if we direct Cas9 to the promoter region, then the transcription factors that come with Cas9 to that region then express that gene in high levels. So Jason and Maggie were really driving that project from WeHi, from Marco Herold's lab. And so we have these shown here as a proof of concept that we were able to use that activator system as well. And so that's currently under review. And also there's also like a nice whole genome screen with that. We have another aspect of disease are mutations, like small mutations, like one letter changes. And a lot of these occur in a protein gene called P53, which is mutated in 50% of all cancers. So it's really like one of the master regulators of cancers. Usually it's a tumor suppressor gene, so we really need it because otherwise we get cancer. So that's why cancers have it mutated. And we can use CRISPR-based editing, similar to activation, where we use a Cas9 version which doesn't cut the DNA, but it sort of uses that RNA GPS signal to go to a particular location on our DNA where we would have that mutation. And then because we can fuse another protein like deaminases, we can actually directly change a nucleotide letter from C to T or from A to G, depending on which base editor we use. And we did that as proof of concept as well. So, and actively now we're trying to use that system for similar whole genome screening, where we, instead of knocking genes out or instead of overexpressing genes, we specifically introduce all these different mutations. And I'm focusing here on P53 sort of that's our pipeline in the lab that we would do. But p53 has a lot of mutations in cancer. As I mentioned, they're all in the DNA binding domain of p53. So p53 is a protein transcription factor which binds to DNA and then drives the expression of lots of genes. And so cancers really have mutated that protein because they don't want that tumor suppressor, because they want to become a tumor. And so we can either do targeted screens focused on p53, or we can do like whole genome screens with this base editing technology as well. And sort of summarizing that aspect, where we have these different tools for different sort of purposes, but all sort of aiming to the same cause of finding novel disease targets, if you want. And I think I just have 2 slides on diagnostics just to sort of give a flavor of what I mean with that. So we all we all know that probably, if I would ask you what that is, then everyone would probably say coronavirus. So we have these like spike protein on there. And so we all did a lot of coronavirus tests. A lot of us probably had COVID themselves, but so we can use CRISPR technology to actually have a quick, reliable test as well. And so, as I said, Cas9 was used for genome editing, but then because the numbering sort of gave it away, so you have also other CRISPR scissors, I call them. So for diagnostics, we would use Cas12 and Cas13, and what they have different to Cas9 is this collateral activity. So Cas9 with that RNA GPS system binds and cuts one location. Cas12 and Cas13 also have that RNA GPS system, but once they cut on that specific sequence, they become activated and cut every DNA that they can find around them. So in a cell setting, you can imagine that would chop up our genome, which is not great. But it can be used for diagnostics using sort of this workflow here, where you have if you want to check for viral nucleotides, viral DNA, you can use Cas12, Cas13, program that with the RNA GPS to find a particular sequence on that virus, so that we know it's actually that particular virus. And then it would bind if it finds it, and it would cut it, which then makes the enzyme to become super active in a collateral way. And that can be used by us if we have a reporter molecule. So we add reporter DNA or RNA into the system as well. And as soon as when our CRISPR scissor cut the target, it also cuts the reporter. And then what then happens is there's like fluorescent signal coming from the reporter that we can measure. So if we get fluorescence measuring, and so I've sort of summarized that here in our workflow, if we have to put all the reagents into a tube, and then we have different Cas12, Cas13, depending on if it's RNA virus, DNA virus, or other sort of pathogen. If it's really detecting the target organism that we want to target, then it becomes active and cuts our reporter, which we can either measure in a fluorescence sort of way with the machine, or ideally it's also sort of in a little test strip, or similar to what we know from those rat tests. And so CRISPR lends itself in a really nice way to have a rapid and reliable point of care diagnostic testing method. And this is something I'm sort of looking at doing at Sturt University, where I'm now. And so just summarizing, so CRISPR used for you mostly use it for genome for disease modeling using genome editing and finding, like doing all these big screens in the haystack to find novel disease targets, using it for diagnostics, and also just going back to that CRISPR 101 online learning course, because I feel like that was a lot of content to digest. So really feel free to go back to that resource that we just published. And yeah, thank you very much. So this is my acknowledgement. So lots of people from way high, yeah, Charles Sturt, going back to reconciliation, Jinjamara Vinangana. So that's sort of Charles Sturt's mantra to create a world that's worth living in. And so yeah, that's virtual language. And yeah, with that, I just leave it at that. And any questions, I'm happy to answer, or send me an email as well.